Hi, and welcome to our second mind map this week. And now we're going to be revising the topic on molecules. So we're going to be looking at the major types of biological macromolecules that are found within cells. So before we get started, the first thing that you need to do, if you haven't done so yet, is to print out the key images that I'm going to be sending you on a PowerPoint and cut those out. And again, you should be making an A3 mind map, so you can either do that in a double page spread of your book, or you can take two pieces of A4 paper together. I'm going to try and cover this whole topic, which is on chapter 2.2 of your textbook. So first of all, let's just kind of orientate ourselves. Where are we here? Well, let's divide up this grid. Okay, so first off, we've got this area up here. So I'm going to kind of just section off this part here. This bit up here is going to be about water. So let's label that. Then down this region, sort of around about here, moving down to about here, is going to be about carbohydrates. So let's put that there. Up here, we're going to be looking at lipids. Just this top segment. So I'm going to move that across there. So this is going to be lipids. And this whole section down here is going to be about proteins. Okay, so if you took a cell, a typical mammalian cell, well, first of all, about 70% of it would be water. So this is just rough estimates, just to give you a bit of orientation. 70% of a typical cell is water. So what would be the rest of it? Well, the rest of it, if you dried out the cell and you measured the different components, most of the remainder of the cell would be protein. So proteins are around about 18% of a mammalian cell. Lipids, so that's phospholipids and other lipids, would account for around about 5% of the cell. So, you know, that could be all the membrane, so the nuclear envelope, the extra, so the phospholipid bilayer on the outside of the cell, the cell surface membrane, also the endoplasmic reticulum, the rough and smooth endoplasmic reticulum. And then carbohydrates in a mammalian cell would be around about 2%. In fact, it's probably a little bit higher than that, actually. That figure 2% is just for the polysaccharides, but we're also talking, there's glucose as well. So I'm going to have a bit of a guesstimate here and maybe say that that's about 3 or 4%. Let's say 4%. But of course, those figures I got from a mammalian cell, and in a plant cell, this figure would be way higher because plants use carbohydrates as structural elements as well as a respiratory substrate, i.e. as well as things just for energy. Okay, so let's now zoom in and let's talk about water in a little bit more detail. First thing you need to know about water is that it's H2O, of course, and that it is a polar molecule. So when we label a water molecule, we label it with these symbols delta plus and delta minus. And that's because the covalent bond between the hydrogen here and the oxygen here is kind of unequal. Oxygen is more electronegative, which means it pulls the electrons towards it, giving the covalent bond a bit of an uneven character. So it's because of these this uneven charge that water has many special properties. So when we put lots of water molecules together, I'll label these over here. We could say this is delta minus, this is delta plus, this is delta minus here, this is delta plus. We get the formation of these special dotted lines on this diagram here, and these things here are hydrogen bonds. Okay, so those are really what give water all its special properties. So hydrogen bonds are weak in isolation. So one hydrogen bond is a weakish bond, but when we put lots of them together, they add up to be quite a big force, and it changes the properties of water. So what are some of the key properties of water that hydrogen bonds are responsible for? Well, let's list a few down here. Well, let's say properties. Okay, so properties of water. Well, it's a great solvent. That's not really because of the hydrogen bonds. It's more because of the polarity of the molecule. So, for example, if I I could maybe have a sodium ion up here. Let's say if that was a sodium ion there, then the delta minus the oxygen is going to orientate itself to kind of face the sodium. So you could imagine lots more of these water molecules. Like here would be one up here, and here would be one over here. They would all sort of orientate with respect to the sodium, which means they can dissolve ions quite easily. But equally, we could have a Cl minus ion, let's say over here, and that would be sort of orientating close to the hydrogens here. So it is a very good solvent, especially of polar substances. Also, we can see that these hydrogen bonds here, they are causing the water molecules to stick together. So we have something called cohesion. Cohesion means the water molecules stick together. We can see that here. Similar to cohesion, something a bit similar is called adhesion. So adhesion is the water molecules stick to other things. Quick demonstration of properties of water. Piece of tissue, glass of water. If I put tissue in the water, you know what's going to happen. The water is going to be drawn up. And that's actually because the water molecules are kind of sticking to the sort of the substrate, this tissue paper. And the water molecules, because of that, cohesion and adhesion are drawn up the paper. That's important for things like xylem, for example. And it gives rise to lots of properties of water. Okay, what else have we got? Also, when these hydrogen bond networks form on the surface, they're responsible for surface tension. Which is how this pond skater here is sitting on top of the pond. Another thing, when water cools down, when it becomes ice, the hydrogen bond network between the molecules forms a regular pattern. So this is an odd one. So ice is less dense than water. So what does that mean? Well, that means that ice floats and therefore insulates the water underneath. So if I was to try and draw, I don't know, like a lake here. Okay, this is my lake. Okay, this is a lake in the deep winter. Here's the water underneath. Right, all this water underneath stays liquid water, and then how am I going to draw ice? I'm going to draw ice in kind of grey. This layer of ice on top actually insulates the water underneath. So fish can still sort of swim around up here, and up here it's frozen solid. That means that you get this sort of habitat underneath the ice is maintained. So it provides a habitat. Okay, surface tension. So let's just highlight those key ones, just so they stand out a bit more. We've got surface tension. We've got the fact that ice is less dense. We've got the fact that it's a great solvent. We've got cohesion. We've got adhesion. Have I missed anything out? Yes, I have. One more thing is that water has a very high specific heat capacity. So where am I going to put that? I'll put it up here. So high specific heat capacity. And that means that water maintains its temperature, whether you supply it with a lot of energy, you take a lot of energy away from it, it kind of keeps a relatively stable temperature, and that's good for creating stable environments. So either stable environments under the ice like this, and it also means that it's easier to control our body temperatures because you need to put in a lot of energy to change the temperature of a human body because it's 70% water. So high specific heat capacity is another one. Also, similar to specific heat capacity, we've got another one which is, I'll put it over here, high, it's called latent heat of vaporization, which means it takes a lot of energy to evaporate water and turn it into a gas. That is biologically significant because it means that we can use it to cool our bodies. So sweat, when it evaporates, takes a lot of energy away, and also just means that things like ponds and puddles and stuff are kind of likely to stick around and be a habitat for a large amount of time. I think that's everything I've got. And the other thing is, of course, that it can be a reactant, and we're going to look at the way that water can be a reactant up here. So let's just highlight those two other extra ones. So we've got the latent heat, high specific heat capacity. Okay, so those are all the properties of water. Kind of branch them out everywhere from this central diagram around here and everywhere. All these are properties of water. And most of these properties are down to these hydrogen bonds. Okay, so what about this up here? So the last thing I said is that water can be a reactant. And the main thing that you need to know about water is it's a reactant in two, well, it plays a role in very, very important type of reaction called a condensation reaction or a hydrolysis reaction. So really what this shows here is that if we have two molecules, try and zoom in a teeny bit more, two molecules each with this OH group, okay, this hydroxyl group here. Now what can happen is that the two hydroxyl groups here come together and we get a condensation reaction. So what happens there is this H and this OH here, I'll kind of highlight that like this, they join together and they form water. So I can keep that same roughly color scheme. So when these two joins together, water will be formed as an extra product, H2O. The two molecules there will link together and will now no longer be separate. Let's just color code that so we can kind of see that more easily. Let's do that with that one. Okay, so now these two molecules will be linked. So molecule 3 is a join of molecule 1 and molecule 2. And then if we wanted to split this molecule 3 back into its original constituents, we'd have a hydrolysis reaction. Oops, and we'd have to get rid of that. We'd have to supply water into that reaction to do hydrolysis. Okay, so the two things that we need to know, two sort of different types of reaction, is condensation. Condensation reaction joins things together, and then we have hydrolysis reactions split things apart. Now, it's worth noting that condensation reactions, it's worth noting that hydrolysis reactions, sometimes in biology, in kind of practical biology, you need to split molecules by doing hydrolysis. The example I'm thinking of is sometimes you need to split a disaccharide into two monosaccharides using hydrolysis, and it's not good enough just to put the disaccharide, for example, sucrose, into water. You normally have to use a catalyst. So typically this might need, in a lab, it might need an acid catalyst for that to happen. Okay, so that's key principles: hydrolysis and condensation reactions involving water, either producing water as a product or using water as a reactant. And it's a way of joining molecules together or splitting them apart. Before we move on, I just added a little bit right here, just sort of some of the notes from what we were talking about earlier. Water is a polar molecule. Oxygen is more electronegative than hydrogen. And one of the significant factors about the fact that it's such a good solvent, it's a solvent of polar substances, is that it's useful as a transport medium. For example, human blood, also xylem and phloem, that all those different transport systems use water to send things around a plant or a human. Okay, the next thing that we're going to look at is carbohydrates. Okay, so what are carbohydrates? Okay, the first thing that we need to know about carbohydrates is that they are built of sugars. So where are we going to talk about sugars? We're going to talk about sugars probably down here, in fact. Okay, so sugars. Sugars are the fundamental building block of carbohydrates. So what is a sugar? Well, sugars are sweet. They are crystalline solids. And they have a formula that is CH2O brackets to the N, generally speaking, like that. CH2O to the N. For example, here's an example. If N is 6, then we have this, C6H12O6, and that is glucose. That's glucose, yeah. Probably glucose, could be some other things as well. So that's glucose. We can also have N. So that, in this case, N would be 6. So N is 6 carbons. So N can be 6, and that would give us a hexose sugar. It could be 5, and that would give us a pentose sugar. Or it could be 3, which would give us a triose sugar. Okay, we don't do much about trioses. We do look at them a little bit in year 13 when we're looking at respiration and photosynthesis. But in year 12, we're mainly thinking about hexose sugars and pentose sugars. Now, if you've got your textbook with you, on page, 55, there's information about some different sugars there with some different structures that we need to know. The most important structure that we do need to know is glucose, and it comes in 2 forms. So glucose, C6H12O6, actually there's 2 varieties. Let's drill this in green out here. So let's use blue actually. So right, glucose, 2 varieties, isomers. We've got alpha glucose and we've got beta glucose. Okay, so the way of drawing alpha glucose, you don't need to know the full structure, so I'm not going to draw it here, you don't need to know it. We start with the oxygen there. Okay, oxygen in this position, kind of the top right hand corner of the hexagon. And we go down like this, and we draw our hexagon like that, and we go round, and then carbon 6 sticks up like there. And for alpha glucose, the OH on this carbon 1 goes down like that, okay? And this would be a hydrogen over here. For beta glucose, we start with the same oxygen in the sort of top right corner. We draw our hexagon, like this, but the OH on this one goes up like that. So the key difference is the position of the OH on carbon 1. What do I mean by carbon 1? Well, if you were to start at oxygen and go round as if you were sort of going around a clock face, then this is an oxygen atom. The first carbon atom is there. This point here represents a carbon atom. This point, this vertex here, represents a second carbon atom. This one represents the third. This one represents the fourth. This one represents the 5th carbon atom, and that is the 6th carbon atom. That's going to be important. I'll just quickly do that over here as well. 1, 2, 3, 4, 5, and 6. The ability to specify the carbon atom in the ring is important when we're talking about bonds. You can see it over here, 1, 4, 1, 6, and so on. And it's also going to be important when we're looking at DNA structure because a key pentose, can I fit this down here? Yes, I can. A key pentose that you need to know about, or the two key pentoses that we need to know about really are deoxyribose, which is found in DNA, and the other one is ribose, which is found in RNA. So yeah, glucose here is a hexose. The other hexoses that we can have here, which are important, are fructose, and also we've got beta-galactose. Okay, all right. So those are the fundamental building blocks, sugars. Now, sugars are also called, if they're one ring like this, they are called monosaccharides. Monosaccharides. And that means that they're sort of comprised of one of these hexagonal or one of these ring structures. But they can also be disaccharides. So how do we take a monosaccharide and turn it into a disaccharide? Well, we come back to that condensation reaction that we talked about earlier. So this here shows a condensation reaction, and this here would show a hydrolysis reaction. So if I was to draw what these two alpha glucose molecules will look like if we join them together, you do have to be able to do this. So basically you just draw your two rings close to each other, the little up there. You don't have to join them yet, just draw them close to each other. So there's the oxygen there, round, round, round we go. The math one is a bit bigger than the other. And then this is where the bond is going to form. We just put an oxygen in the middle like that, and then we just join it like this. So both join to the oxygen. If we were to label those carbons again, if you wanted to, that's 1, 2, 3, 4. So this is carbon 4, and this is carbon 1. So we say this is a 1,4 glycosidic bond. So this there is a glycosidic bond that is formed. Glycosidic bond formed between the two monosaccharides, and that gives us a disaccharide. So maltose is the molecule that is produced, and maltose is a disaccharide. And then we can also split this molecule via hydrolysis. Okay, so maybe a little bit more on key disaccharides. I've kind of run out of space up here, so I'm going to put a little table of disaccharides here. Are produced by condensation. I'll just write a few out here, and you'll find this on page 54 of your book if you've got that with you as well, so you could write that down. Okay, so those are the disaccharides that you need to know. There's quite a few, you just need to memorize these. So just quickly run through them. 2 times glucose, 2 alpha glucoses together, this molecule here is maltose. If we have a glucose and a fructose together, we got sucrose. That is the sugar that we put, on food, okay? Sucrose is the sugar that we find in fruits. We put it on food, so put sugar in your tea, it's sucrose, it's a disaccharide. What else? 2 times beta glucose together, we get something called cellobiose. It's a bit of an old one, that one. And this one is significant. Beta galactose plus alpha glucose produces lactose. Now, some people are lactose intolerant, and that means they don't have the enzyme to break up this disaccharide into the 2 monosaccharides. So we need a specific enzyme called lactase can break the 1,4 glycosidic bond between beta galactose and alpha glucose. Okay, so I'll just write lactase enzyme. You can fill in the rest. Okay, so that's everything about sugars, monosaccharides, disaccharides, condensation, and hydrolysis. Now we're going to move further out and talk about what happens when we join more than two monosaccharides together? We don't get mono, we don't get di, but we get polysaccharides. Okay, so that's where we're going next. Okay, so now we're going to move on to polysaccharides. So polysaccharides are just lots and lots of these monosaccharides joined together. And most polysaccharides that we look at in A Level Biology are polymers of alpha glucose, with one key exception, which we're going to look at later. So all of these over here are polysaccharides. So let's label this polysaccharides. So a polysaccharide is a polymer of monosaccharides, and I think typically in these polymers we've got sort of in the thousands of monomers joined together. And there's three polymers of alpha glucose that we need to know about here. So the first one up here is amylose. The second one down here is amylopectin. And the last one is glycogen. Okay, so amylose, as you can hopefully see here, has got only alpha 1,4 glycosidic bonds. Okay, so 1,4 glycosidic bonds between carbon 1 and carbon 4. So just like we've shown here, this 1,4 is the type of bond that is present. Now, what else do we need to know? Well, for amylose, we need to know that this is a component of starch. And when we have this chain, because it's alpha glucose and they all join in a line, there's a slight curve. So we can imagine if that continued on, it would just curve around like this. So that means if we zoom out, the structure of it is actually a spiral. Forms a spiral. And the test for amylose is iodine. So the molecule iodine, I don't know what colour I'm going to draw that. Let's make it kind of brown blob. So iodine kind of actually fits in that spiral, and when it fits in that spiral, the iodine changes from brownish to bluish blackish. So that's why we use iodine to test for starch, because the atom of iodine actually fits within that coiled amylose chain. Haven't really got much space to write that, but maybe you should write that down on your mind map. Okay, what else? Amylopectin is the other component of starch. Okay, so this is another component, smaller component of starch. And we should say that both amylose and amylopectin together make starch. Maybe I can sort of show it like that. So starch is made of those two. And starch is, of course, found in plants. So it is the store of energy in plants. Now, amylopectin contains two different types of bonds. It's got 1,4 glycosidic bonds, and it also has branches, which are introduced by these 1,6 glycosidic bonds here. Maybe I'll colour that one actually a different colour. Let's do that, and let's colour that like that there. Okay, so that's 1,4 and that's 1,6. All right. What else? Glycogen is found in mammals. So animals, not just mammals, sorry, animals. And it has both 1,4 and 1,6 glycosidic bonds, both of them, but it is more branched, okay? Now, what's the significance of all this? Why do we use these polysaccharides? Okay, so the reason we use these polysaccharides as energy stores is, where am I going to put this? I'm going to put this down sort of here. So glycogen's an energy store, so is amylopectin. and so is amylose. They're all energy stores. The reason is because they are compact, okay? You can fit a lot of glucose into a tight space if you stick them all together in a polymer. Okay, so compact, that's key. Also, they are non-soluble. Now that means that you can store lots and lots of glucose molecules together as a polymer in a molecule of one of these polysaccharides inside the cell without it affecting osmosis. So because they're non-soluble, doesn't affect osmosis. In fact, let's make that better. It doesn't affect water potential. Remember this symbol there for water potential. And the final thing is that they are readily hydrolyzable. I'm going to write that over here. Move across slightly. Readily hydrolyzable. That means hydrolysis, remember, is breaking a larger molecule down into its smaller parts. So readily hydrolyzable means you can kind of chop it up from the polymer back into the monomer, so the cell can have access to sugar for respiration, glucose for respiration whenever it wants to. And the main thing about glycogen is that it is more branched and it is more readily hydrolyzable. So maybe I'll sort of do a little side note off of this more branched here. That means that has more ends. So if you, a molecule of glycogen is kind of like this. It has a central core and then it has, so many ends branching off like this. And each of these ends is where an enzyme can bind and start kind of breaking down, chomping a bit like Pac-Man, chomping down the chain, liberating those alpha glucose molecules. So more ends for enzymes to bind and then do the hydrolysis. Okay, and that's significant because animals need to get energy released more quickly because we might need to run. So if you're running a race, you probably burn up the glucose in the first few minutes of running a race, and then your body starts breaking down glycogen. So you obviously need to be breaking down glycogen to provide that blood sugar, which will then power your muscles, and those stores of glycogen will last maybe a couple hours depending on your level of activity. Okay, so those polysaccharides as energy stores. So amylose, amylopectin, glycogen, very similar molecules really. Well, amylopectin and glycogen are very similar. And they're all used as energy stores. Polysaccharides as energy stores. Now there is another polysaccharide, which is cellulose, and that's quite different. Okay, so this one's cellulose. Now the main key thing about cellulose, which I'm sure you remember, just move across here, is that it is a polymer of beta glucose. Now, you really wouldn't think that it would make such a big difference. Remember, alpha glucose is this over here, and beta glucose is this down here. So it's all about the orientation of this hydroxyl group on carbon 1. So because the hydroxyl group is facing up in carbon 1 on beta glucose, and the hydroxyl group on carbon 4 is facing down, I'll actually draw that in over here. So this one over here always faces down in alpha or beta. I can add that over here as well. This means that every time you add a beta glucose to the chain, you need to flip it 180 degrees. So if we focus on the carbon 6, the direction of the carbon 6, and I'll highlight that here. Let's highlight it in green. So here's the carbon 6 there. 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 So you can see each residue is flipped 180 degrees. Up, down, up, down, up, down. And if we see that, the oxygen, we could say the same thing. Up, down, up, down, up, down. Now, why does that matter? Well, it means that we have a very straight chain. Now you may remember, if I just point you back over here, that when you join them in a chain where all the carbon 6s are facing up, I'll just label those here, like there's 1, there's 2, there's 3. If they all face up, it makes the chain curve slowly, which gives it the spiral shape. That isn't present in a polymer of beta glucose, so very straight chain. Now, because of that very straight chain, this chain here, we can kind of, let's zoom out from it, and this shows you, you can see those hexagons there if you look really closely. That is the molecule kind of viewed from a distance. Now, each of these cellulose fibrils, so that there is one cellulose chain, actually. Each of these cellulose chains can lie next to several other cellulose chains in a perfectly straight sort of macromolecule. So we get several hundred chains forming together like this. So, maybe you have 100 chains or something like that. Like that, lying together. And in between those 100 chains are hydrogen bonds between the chains. So if I could draw those in, that would, those would be sort of like here, there would be a hydrogen bond to another one below, and there'd be a hydrogen bond up there and down there, and in other places as well that I'm not drawing. There'd be hydrogen bonds to adjacent parallel chains. So hydrogen bonds between chains, and I could even put the word parallel in there, actually. Let's just kind of add that in there, between parallel chains. So that makes this molecule very strong. Now, we don't just stop there. We take these take these 100 chains, and we bundle them together into what's called a microfibril. And we bundle lots of microfibrils into macrofibrils. Macrofibrils, that's it, right. And it's these macrofibrils that make up the cell wall. So actually, if you look at a cell wall from far away, you can actually look at it with an electron microscope, and you can see these macrofibrils. And they kind of crisscross, a little bit like my fingers doing like this. They crisscross across the outside of the cell, which means that it gives it very strong structural support, but it's still permeable because water molecules can go between the gaps of my fingers, like so. So that those macrofibrils make up the cell wall. Cell wall, strong, high tensile strength, which can resist the kind of pressure of osmosis. And if we were to look at kind of the cell wall, these macrofibrils are kind of crisscrossy like this across the cell wall, which means the cell wall is still permeable, but it's very tough. Okay, so those are the key polysaccharides that we need to know about. All right, let's zoom out a little. Okay, so we've zoomed out here. Now just quickly review what we've done. We talked about water, we've talked about carbohydrates, what is a carbohydrate, sugars, monosaccharides and disaccharides, and we've also talked about polysaccharides. So we're more than halfway there. I've just got two other classes of macromolecule to look at. So let's now move on to lipids. Okay, so now we're on to lipids. So lipids are another type of macromolecule. So that means they're a large molecule, just like carbohydrates are macromolecules. So lipids are macromolecule, but they are often not polymers, okay? So that means they're not made up of a repeating chain. So what does it mean to be a lipid? Well, lipids are, they tend to be alcohol soluble. So they dissolve in alcohol, but not water. So the reason, dissolve in alcohol soluble. So they're alcohol soluble, but not water soluble. And the reason that they're alcohol soluble is that they have, they're non-polar, generally speaking. And they have lots of C and H, and they have little oxygen. Lots of carbon, hydrogen atoms, little oxygen atoms. So that's what a lipid is. Now, lipids are generally formed through condensation reactions. So here we can see a condensation reaction between the two different parts of a lipid. So here we have, I'm just going to highlight these molecules. Let's highlight this up here in blue. So this whole thing here is glycerol, okay? And this here is a fatty acid. Okay, we can join them together by a condensation reaction. So we're familiar with these condensation reactions before. So this is condensation. And this would be hydrolysis. Now here we have a glycerol molecule and a fatty acid joined together to produce one. This bond here is an ester bond. So, and we'd also produce one molecule of water. So that's just one bond. Now, what we can actually do is we can do 3 condensation reactions. We can attach a fatty acid to this OH here. We can attach another one here, and we can attach another one here. So this molecule over here, this is the result of that. And we call it a triglyceride. So it's 1 glycerol and 3 fatty acids that have been joined. If I highlight again in those colours, got fatty acid there, another fatty acid there, fatty acid there, 3 of them, and we've got our glycerol molecule here. Okay, so glycerol, 1, 2, 3 fatty acids there. So triglycerides are basically fats used for storage. But they can be different. We have different fats for storage because of the different fatty acids. So fatty acids can be very different. So let's do a little bit on that here. The 2 major differences in fatty acids are the types of fatty acids. We can have saturated and we can have unsaturated. Now, saturated fatty acids have no carbon-carbon double bonds. And unsaturated, well, we can have mono unsaturated, or we can have poly unsaturated. So polyunsaturated fatty acid has lots of carbon double bonds, and mono will just have 1. Now, why is that significant? Well, let's look at the reason that's significant after we looked at this molecule. So this molecule here is a very special type of lipid, which is called a phospholipid. Now, phospholipid, yeah. So they form the bilayer, the membrane, okay, phospholipid bilayer. Now, phospholipid is very similar to the triglyceride, except it has 1 fatty acid here and a second fatty acid here, but there's no third. Okay, there is not a third phospholipid, and that is because in place of the 3rd fatty acid, we put a phosphate group. So let's just use a pen and let's make a different color. So this is the head, but let's add in a phosphate group. So let's pretend that we've got a P, and the phosphate group is like a phosphorus with oxygen, and it's like that. Okay, and it's very charged. It's highly charged group. So that means the whole of the head region is very, very charged. Let's highlight that in what color? Let's highlight in orange. Okay, it looks like this. Okay, so this whole head region here is highly charged, including this phosphate group I've added in there. Okay, this is a very charged group. This is a very non-polar, and this is very polar. So let's label that again. We've got a polar region and a non-polar region. So the non-polar region is hydrophobic, and the polar region over here is hydrophilic. So I'll just add that over here. Head equals hydrophilic. So we've got hydrophobic and hydrophilic. So that's weird. If we have the hydrophobic and hydrophilic together, we've got a molecule that one part wants to be in water and one part doesn't want to be in water, which we call amphipathic. Amphipathic. So amphipathic, which basically means it's in two minds almost about itself. So how does it solve the problem of wanting to be part in water and part not? Well, it lines up like this. We have a head, and then we have the tails, and we have the head, and we have the tails, and we have the head, and we have the tails. Now, let's zoom in a bit more. I talked about saturated versus unsaturated. Unsaturated, so far all these are saturated fatty acids I've drawn on these phospholipids. Now, unsaturated fatty acids, I'll change colour, look like this. Okay, so when you introduce an unsaturated fatty acid, it means the neighbouring phospholipid can't get as close to it, okay, because of this kink in the molecule. So that means that the phospholipids are spaced further apart. Okay, so by introducing unsaturated fatty acids into a membrane, you make it more fluid. The phospholipid molecules can't pack tightly together like so. So fats which have a higher concentration of unsaturated fatty acids, so I'll just put that on here, unsaturated fatty acids. So fats with a higher proportion of unsaturated fatty acids tend to have a lower melting point. If we're talking about a physical fat, so for example, if you want to put a little example of that, like olive oil. So olive oil is a liquid at room temperature. You know, other vegetable oils as well. Lower melting point. And if it's in a membrane, so the membrane is more fluid. If we're talking about the fatty acids in a membrane is more fluid. Okay, so that's the main difference between saturated and unsaturated. Unsaturated have these kinks in the tail, like so. It's bugging me, this membrane is not complete, so I'm just going to complete this membrane. It's going to include all phospholipids with saturated fatty acids on the inside face of this membrane. Okay, well, there's one little molecule which we almost have removed the room for. This molecule down here, this lovely molecule. Now, this is called cholesterol. I'm going to use black. No, I'm not going to use... Let's use blue. So, cholesterol. Now, cholesterol, let's try and add it in here, actually. Cholesterol is a planar molecule, and it kind of fits... This is a bad drawing of cholesterol here. It fits in the membrane like this, and what it does actually is it regulates the fluidity. So it's a hydrophobic molecule, which means it fits in amongst the tails of the phospholipids, and it regulates membrane fluidity. That's the key phrase. So it's too big. Regulates membrane fluidity. Okay, fits in there, regulates membrane fluidity. Also from cholesterol, I talked about it in the previous mind map. It can be used in steroid hormone synthesis. I'll just write steroid hormone synth. Okay, so that is lipids. One thing that I should say about triglycerides, which I almost haven't remembered to do, is you can pack a lot of energy in. Loads of energy in a triglyceride, partially because of the high C carbon and H content. Okay, so when you respire fats, you'll have to figure out somewhere to put this on your mind map. I've run out of space. When you respire fats, you combine carbon atoms in the fat with oxygen, and you combine hydrogen atoms in the fat with oxygen. And in doing so, you make carbon dioxide and you make water. So you need to breathe in a lot of oxygen to respire the fats, but there's a lot of energy in the fats because you've got carbon and hydrogen atoms almost only. Now, when you're respiring a carbohydrate like a sugar, C6H12O6, there's a lot of oxygen already in there. So that's kind of wasted space in a way, because when you can breathe in the oxygen from the air, you don't really need it in the molecule really for the energy. You just want the carbon and hydrogen. So fats are more energy dense than sugars. Okay, so that's pretty much everything about lipids. And we just got one more key molecule to look at, and that's proteins. Okay, last thing to look at is proteins. Now, proteins are a macromolecule that comprises about 18% of the cell, and they include things like enzymes and also structural proteins like collagen, or transport proteins like hemoglobin in a red blood cell. So proteins can have a really, really huge different variety of structures. This is the reason why. They're built of something called amino acids. So this is an amino acid here. And they're called amino acids because they've got two groups. They've got, two functional groups on them. We've got the amino group here, which is this NH2 group. Let's zoom in a little, actually. That's better. We've got the NH2 group, which is the amino group, and they've got this carboxylic acid group here. So they can act as both an acid and a base. So proteins have a buffering ability. Now, amino acids, they come in 20 different varieties, and they are different via this R group. Okay, so this is the R group, and there are 20 different R groups to give 20 different amino acids. Now, some of these amino acids might be hydrophilic, some of them might be hydrophobic. So let's just sort of do that. We could do hydrophilic, they could be hydrophobic. A few of them even have a charge on them. Or they may be able to form covalent bonds. For example, if it's sulfur, sulfur and sulfur can form a covalent bond outside of the main chain. So when we join amino acids together, we have a condensation reaction. So this is a condensation reaction. We have amino acid here and amino acid here joining together. Now let's highlight the atoms that are involved in that condensation reaction. Let's zoom in a bit more. That's better. Yeah, here we go. So the atoms that are involved in that reaction are really this hydrogen here and this OH here, and these kind of atoms there join together to form water, leaving us with a leaving us with a peptide bond. So let's draw that out. So you can see this amino acid here represents this one, starting at this central C. So I have to draw a line across. Then I bond to my carbon. I've got a double bond up to my oxygen there. And then I go straight across to a nitrogen there, which bonds to that carbon, and this nitrogen's got one hydrogen there. So this bit here, which I've drawn in black, that is actually the peptide bond. So maybe if I highlight that in yellow. And it's that whole group that we consider the peptide bond. Now, actually, I haven't really talked too much about food tests, but I'll briefly mention the food test for proteins. The test equals Biuret reagent, and it goes blue to, let's do it with the ink color, blue to a kind of purple. Okay, and it only tests for the peptide bond. So the Biuret reagent will not detect just amino acids floating around on their own, but it will detect peptides. Okay, so when we put proteins together, so when we join amino acids together into proteins, we talk about levels of structure. Okay, so let's talk about those structures. Move across a little bit here. So first of all, we have, what color am I going to do this in? Let's do red. First of all, we have, oh gosh, sorry, that's too big. Okay, first of all, we have the primary structure. And the primary structure is really just the sequence and number, plus number, let's write NO for number, of amino acids. Alpha is short for amino acids in biochemistry most of the time. Sequence and number of amino acids. So for example, I could draw, let's try and draw, is that going to work? Yeah, okay, so if I drew something like this, oh gosh, hang on a minute, that's massive. No, I want that one. Okay, like that. And what I'm trying to show here is I just have some blobs. And these are, these are my amino acids in a sequence, okay? Each color representing a different amino acid. So that would be a way to represent amino acids with the peptide bonds between them. So that is the primary structure of a protein. Now, once we go past primary structure, obviously we're going to get to the secondary structure. Now, the secondary structure forms when the amino acid chain starts to coil and fold on itself. And really you need to know about two forms of secondary structure, two secondary structure types. So let's just talk about them. We've got the alpha helix and the beta, sometimes called beta pleated sheet. And it's formed by a coiling of the peptide chain. So alpha helix is, well, it's kind of a helix like this. The beta pleated sheet is kind of a folded sheet like this, and then you get a bit of a hairpin, and then it folds back, and then you get a hairpin, and it folds back like that. Okay, so that's a beta pleated sheet, that's alpha helix. And the one thing you might need to also know about the secondary structure is typically it's stabilized by hydrogen bonds between sort of this oxygen here and hydrogens in the main chain. Hydrogen bonds between CO plus H atoms in the main chain. So it's not to do with hydrogen bonds between the R groups, basically. Okay, secondary structure. Right, primary structure, secondary structure. What about tertiary? This diagram here represents tertiary structure. Oops. Tertiary structure. Now, tertiary structure is a further folding and coiling of the protein chain, or the polypeptide chain, caused by now interactions between R groups. We've got a bunch of different types of interactions, so let's label those. Let's label those in green. We've got ionic interactions. Okay, so here's an R group which has got a negative charge, and this R group's got a positive charge, and there's an ionic interaction there. We've got covalent bonds. This is a disulfide bridge, okay, between two cysteine residues. So the amino acid with the S here is a cysteine. Cysteine amino acid has the sulfur as its R group, and it can form a covalent bond to another cysteine. This would be another cysteine over here. What else have we got? We've got hydrogen bonds. Here's another hydrogen bond. And if we're talking about tertiary structure, this would be a hydrogen bond between R groups. And we've also got hydrophilic and hydrophobic interactions. So in this diagram here, this central orange region is amino acids with hydrophobic R groups. And they arrange themselves so they're inside the protein, sort of sheltered away from the aqueous environment, which is water around the outside. And the purple outer bit is amino acids with hydrophilic R groups. Okay, you do sort of need to know which bonds are stronger and which are weaker. So in terms of the bonds that are the strongest, ionic bonds and disulfide bridges are the sort of strongest types of interaction that hold a protein together, and slightly weaker are the hydrogen bonds and then the hydrophilic and hydrophobic interactions. Okay, yeah, so proteins that have a very stable structure, potentially very stable at high temperatures, well, maybe they potentially have a lot of ionic interactions that hold them together. Maybe they have some disulfide bridges that hold that structure together. So maybe an enzyme that works at high temperature might have some more of those interactions to stabilize the structure. Okay, well now we also have a quaternary structure. Pause. Resume. So quaternary structure, let's write that out, quaternary, really is talking about when we have multiple polypeptides. So we call the whole thing the protein. So we really talk about polypeptides when we're talking about kind of sub-proteins within a larger protein. We call them polypeptides. So that means that, here's an example of a polypeptide, okay? So it has 4 different polypeptides, okay? Let's call them alpha 1, alpha 2, beta 1, and beta 2. So in this protein, which happens to be hemoglobin, it is comprised of 2 alpha plus 2 beta subunits together in a tetramer, okay, so i.e. 4 of them together. Therefore, it has a quaternary structure because it's 4 polypeptides. Each polypeptide would be individually translated at the ribosome, and then they're stuck together potentially in the Golgi apparatus where proteins are modified. So this protein has a quaternary structure. Also, this protein is something called a conjugated protein. So a conjugated protein contains other types of chemical group that are covalently bound, and they're always there. They never leave the structure. They're a permanent part of the structure. So this protein here has 4 heme groups. So each alpha and each beta subunit has a heme group in the middle. Let's label that. So that's this one here. And there's one there, there, and there. So it has 4 heme groups. And the heme group, just a little bit more about it, is something called a porphyrin ring structure. Contains Fe. Is it 2 plus or 3 plus? I'm not really sure. Let's say 2 plus, but you might have to fact check that. I'm not sure. Contains Fe2 plus ions. And it's these Fe2 plus ions that binds oxygen molecule. Okay, so the heme protein actually combines 4 oxygen 2 O2 molecules, so 1 on each of its heme groups. So now proteins can form so many thousands and thousands of different types of structure because of those amino acids and the variability there. So let's just do a tiny little bit quick maths about the variability. Let's put up here. Okay, if you have 1 amino acid, you can have 20 varieties. If you have 2 amino acids, you can have 20 times 20. So 20 choices for the first position, 20 choices for the second position. You could have 20 times 20 variations. That's 400 variations, or 20 to the power 2. If you have 3 amino acids, you could have 20 to the power 3. If you have 4 amino acids, you have 20 to the power 4. Okay, typically a protein might be, let's say, 1,000 amino acids long. So you have 20 to the power 1,000 different possible combinations of amino acids. I'm not even going to try to calculate that number. but it's probably more than there are grains of sand in a beach. If you're into maths, you can try and work that out. It might even be more than the number of atoms in the universe. I think it's a pretty big number. So that gives proteins a huge variety of structures and flexibility, and that's why they can do a lot of different roles, from transport proteins to antibodies to enzymes, all sorts of things. Okay, right. We've finished. We've made it through the molecules. Now, one thing that we haven't really talked about, just the last final bit, stay with me, is tests. Okay, now I have no idea why I'm going to put this on my mind map, so I'm not even going to write it down. I'm just going to describe them, and you can figure out where to put them. So, we talked about carbohydrates, okay? Test for carbohydrate, actually we have talked about that already. Iodine in there, I don't think I labelled it though, so let's label that. I'm trying to write down there, iodine. Okay, iodine is the test for starch, and it goes blue-black. Lipids, we have the emulsion test. Write down something about that, find it in your book, the emulsion test. Remember, lipids are alcohol soluble but not water soluble, so the emulsion test first dissolves the lipid in alcohol, should form a clear dissolved solution. Then you add some water and then it goes cloudy, and that's because the fat is precipitating out, forming tiny droplets which give it a cloudy appearance. And then we talked about the Burette test for the peptide bond. Here gives a blue to purple color change. Okay, so I think that's all the food tests. We talked about starch, peptides, lipids, and that's it. We're done. So spend some time going over that, add some more detail from your book, send me a picture of it on Teams, please. And then the next one we're going to be doing of these mind maps is going to be looking at another molecule, important biochemical molecule called DNA. Okay, thanks very much. Bye-bye.